you guys won't want to see one more such a small technical talk on a particular algorithm. So I dare myself to do something totally different. I want to entertain you, to prepare your appetite for the lunch with something that is kind of crazy and not what you often see in this context. I'm going to present a rather high-level, broad stroke a project, a description of the project that I've been very excited about without diving into the details. And as also Mike mentioned yesterday, with enough young experts and scientists here talking about all sorts of details. And here I'm going to kind of point my focus on the problems, the bigger problems that we might find new opportunities, or maybe to find that it's total crap and we should move away from that. So don't be afraid of just totally trashing what I talk about. So the title is kind of big, and it reflects my background as a computational scientist, but also a biologist. I want to reflect kind of the past from X-ray crystallography, which was a fundamental a cornerstone innovation in biology, all the way to what we saw a few years ago, AlphaFold, and then now the topic of generative biology. And I put a big title there, Renaissance of Empiricism and Connectionism in Biological Science. The title because I used to give this talk once only before in the Royal Society to kind of, again, maybe stimulate some interesting debates. So, right, so data-enabled, big data-enabled foundation model, it's booming. We saw all these curves. In fact, I think we need to add a few more points now with the publication of the Gemini and a few other latest new models. The important thing to remember is that just in a matter of a few years, 2 or 3 years, we are seeing orders of magnitude increase in the size of the model. And it is defined against all the mathematically inspired laws that we used to describe sample complexity, overfitting, and all that. Basically, the model keep growing. And also, it appeared to be delivering newer utilities and functions, right? One thing, of course, is generating everything. It is really generating text, image, plans, a lot of other things. And when you prompt it properly, okay, when you kind of put a text there, maybe you put an image there. And naturally, if I put myself as a very, very ignorant and naive user, I would ask the following: what if I put all this biological stuff, that we have been working on for so many years, also into, say, a foundation model? If you don't like the word foundation model, you can use pre-trained large models. Fine. Basically, it's a big model that is trained on the big data, which now is biology, or maybe a personalized health record data. And can we also get something, as interesting as, say, a personalized medicine, or maybe even just a daily, healthcare management, right? That's basically what we're kind of getting now from the large language model. You prompt, and then it comes back to you with something quite interesting, at least to a outsider standpoint. This is very different from the now practice of drug design and drug discovery. What we are doing now, still now, for the past half a decade or maybe longer, is that you started from a astronomical number of possibilities, compounds, and ingredients, and then you go through multiple stages of literally sifting. Okay, the sifting is by designing the experiments without actually knowing the mechanism of how they work. You literally just sift, right? And we have names for all these trials and phase 1, phase 2, and regulatory approval and so forth. So at the end of the day, if you are lucky, you will get one thing out of it. That process usually takes like a decade. And also it takes literally billions of dollars, right? And that's not, the way we design, our other things like a chip, write a program or, build a building. It is not based, in many cases, at least the full picture of biology. It may be based on a little bit about biological knowledges, but it's not, in fact, we don't even know what is biology, for a cell. As a biologist, I can tell you we don't know too much, even though we feel like we know. We invent words like a network, regulatory, circuitry, and all that kind of thing. I tell you, these things doesn't exist physically in the cell. Okay, it's just our way of describing what's happening. Okay, it appears to be a first principle, but it's actually not quite there yet. Okay, you cannot, for example, put up an equation and do some calculation and start to compute the outcome, of a cell response for anything, right? So that's not based on first principle. And so can we replace this whole thing, with, for example, a foundation model? That's kind of the question I've been asking, right? What prevents that from happening in the past? Well, primarily because biological systems, as we all know, it's very complex. It's probably more complex than our language, okay, on which we are, training, for large language models. Look at the total number of atoms, molecules, in their interactions at all different levels, and then perturbation interventions, and then, of course, space and time, all that. It's this myriad of, biological, ingredients and the components are really extremely complicated. And in the past, the way we studied actually is a very empirical approach. Okay, we started collecting data of all kinds. Okay, modern day biology started exactly from a new way of collecting data, which is in here. Okay, in 1950, that era, you use an X-ray to shine, a crystallized piece of molecule. You get their shadow, and then you compute through the Planck equation and the reverse kind of a guess or compute the structure. It is not through first principle. Okay, and then we continue to gather more data. In fact, the whole history of modern biology is about new technologies, of getting data, and maybe some ways of disturbing and interfering or changing the system. We measure gene expression. That was the time when I was a graduate student. It's a big deal, measuring a few thousand genes and put them onto a matrix and do something. One of my first papers with Mike was to do feature selection, in these 20,000 genes and get the informative ones out of it. And then there are all these evolution of new techniques, and now we can actually measure the gene, or maybe we call mRNA, which is a precursor of the protein, in terms of their intensity and the volume in a single cell, okay, space and time specific. But on the other hand, this is very different from we set up a camera and looking a one cell continuously growing across time and space. Okay, once you measure, the cell is gone. You have to do another thing. Therefore, there is a lot of amount of statistics, mathematics involved to even make sense of the data that you collect from actually different systems, and then put them together, and then tell a story, right? And the volume of data right now is actually quite big already. People don't realize that, but we actually have the same type of avalanche of data as we see in the internet about the web, in the text and the image. For example, there are tens of hundreds of millions of sequences, of individuals and species. There are hundreds of millions of single-cell experiments, being documented and reported, and across the spectrum, we are talking literally at the order of hundreds of millions, if not bigger. And this number is growing very fast. And again, there are techniques, many of them well statistically and mathematically motivated to analyze all these. So here are just examples of that. And that basically is how biology is done now. Okay, it's very much like in our linguistic world, we put a particular dialect, maybe in the special topic, let's say, classical literature, and then hand it to a lab, and they basically study, everything there. And then another language, another topic, and it's very siloed. Okay, and for a good reason, because the classical approach, like this one, we use for cell biology, were kind of based on an implicit assumption of small data. Okay, sorry, I think I'm pointing to the wrong direction. Yeah, and therefore, we actually, in fact, our very educational system, in the university was established to honor such a science. For example, we established a department of cell biology. We also have departments of molecular biology. We have the department of microbiology, and then all the way up, you have plant science, animal science. Then in the hospital, every organ of your cell becomes a division. You have a division of a liver, a division of a brain, and so on and so forth. It's all kind of cut apart. And imagine that when a patient goes to a hospital and he is sick, all over, you can imagine what happens. The doctors from different divisions were called together. Even to sit in the same room and speak the same language is already a big deal. But at the end of the day, it's actually important to realize that we actually are looking probably at one organism, one instance of an organism, which is a human being or a cell. And the DNA, the protein, the cell, the tissue, everything actually is just a different granularity of the same thing. Okay, that's actually very important. It's the same thing, but it's actually coming from different granularity, right? And now they are studied in a completely or very much siloed way. Okay, in a non-community. So, the natural question, and I think the reason is because of resource limitation and capacity limitation, because we don't have the right tool, for example, to put them together. But now we are in a new era of these large pre-trained models. Like it or not, they show us already a few instances about when you put everything together before you even know everything. Okay, you just put all the languages, all the text instances, everything together. Before you even read it, something interesting happened. Okay, also before we even know why, but it happened, right? So that actually challenged us the question, if there is a way that you at least have the storage of store all data together, and if you have, say, 10,000 GPUs, and if you have that amount of infrastructure and people, are you still going to create 100 departments and divide your machines and data to all of them, or you want to group them together and do it slightly different way? Right. So that's actually what also MBZI is about. The reason I came here and built this school is exactly to see the alternative, which is something not done in established schools and companies, that when you have such a big resource, or maybe a big question, and also when data become abundant, maybe you want to do something differently, right? So that's kind of the gist of this talk, which is, again, very high level. We want to see a way to move away from all this classical approach, which are largely analytical and reductionist. That's actually where we were trained and we are very good at, into something that is a little bit crazy, okay, and less elegant. The LSMs, the transformers, the bigger models, yeah, they are very dirty and ad hoc. But, just like the first microscope was invented, it was, actually really causing people to rethink how they do biology, right? So that's basically, what I want to, maybe speak a few more words. There are two paths in the past, in biological research. It's always been the case. There are people who, come from this highly, rigorous, technical background, championing rationalism and reductionism. And the word there is, let's look at causality. Let's extract first principle. Let's use the law of physics and mathematics, to really do things extremely elegantly, predictively, and provably. Right? And that's actually how we, for example, compute the ellipse. You can actually compute the timing of the ellipse, based on some very simple laws nowadays, and to a great deal of satisfaction. It becomes even a fun thing that on CNN and Fox, you announce, hey, at a particular moment in the United States, you should show up. If you're good weather, you will see something magical. Right? That's a very, very beautiful accomplishment of first principle and reductionism. approach to science. And it really, gets you the satisfaction and also the confidence. But unfortunately, symbolism and the computationalism scale very poorly, especially in the problem of big complexity, such as in biology. In fact, we all know that as a physicist, you cannot even use first principle to compute the very interaction of maybe 3 bodies or 4 bodies, right? And any biological molecule has millions of atoms already. So that path actually isn't getting us to what we want. Then comes the empiricism and the connectionism, which is based on trial and error, data-driven, and sometimes it's a black box. Okay. In fact, the biologists who use electron microscope, or fancy atomic force, they don't know a thing about how it worked. They use it anyway. To them, it is a black box. To the physicists, it's not, but to them it is. But it doesn't prevent them from using it. And also, it generates some kind of understanding, again, very different from there. Okay, the understanding is not about the orbits, of the whole thing, and also, the law in the form of a few equations. It's really in the form of a empirical understanding that is focusing on actionability. You want to do something. So I want to give you the example of how vaccine was discovered. Vaccine was discovered in the day before we know DNA, before we know protein, before we actually know the modern picture of biology and a cell, right? And Pasteur basically did a bunch of experiments. And he found that, when some animals, were intentionally, contaminated or infected with a dead form or a very weak form of the disease or the virus, they actually become immune to the real form, of the disease. I have to take some more time, sorry, for that, right? And the result was repeatable, okay? And also, it became highly actionable. Okay, before you know any why, any genetics, any genes, you just immune or inoculate that guy with a weak form of the vaccine of the antigen, and you're done. And how to treat that antigen is also very well documented. So I call them actionable empirical understanding. You focus on the utility of that. Right, so I guess, I want to pass this page. There's a whole kind of history of the success of empiricism, especially in biology, because maybe biology is so complicated. People just, have to make some progress before you know everything. For example, our very knowledge about embryogenesis, okay? We have a full picture of embryogenesis right now of human and of this little animal called Drosophila. This looks like a movie, right? It's actually not a movie. It's not that you have a little, video recorder looking at the thing being dividing. It's actually stitched together from tens of thousands of pictures taken on different embryos at different stages and so forth, but then they put together. So this is not a causal inference, not a real-time recording. It's literally a reconstruction of the process based on a very strange sampling process. And then, of course, X-ray crystallography, right? What happens is that you basically make a crystal out of the protein, which is already not a native form of the protein because they are crystallized. Then you shine the X-ray, you create all this very complicated form of the shadows. And then you run some programs, which is a complex form of linear or nonlinear kind of deconvolution, of the information, and then you kind of figure out the protein. In a sense, what we see as a protein structure here is not in a rigorous form what we see. We didn't see anything. Okay, it is an output of a computation. Okay, and also an output of the way you prepare the form, the samples. Right. And now what's happening with AlphaFold is just this part is replaced by a program. Okay, not that basically disturbing if you actually know how the original experiment was done. Yeah, so this is basically what's used nowadays in many of the largest models. Layers of tensor operators, right? Starting from some attention based on linear operation over the inputs and some transformed form of the inputs. and then you have an array of those attention heads. Then you have a stack of these arrays of attentions, and you call them an encoder. And then you have multiple encoders arranged in a certain way. You have a transformer, right? And you can also reverse that. If you look at this, I actually when I try to explain this to say a non-scientist, I actually all of a sudden came to the realization that this is no different from the optical or electrical microscope that we use in biology. What is a microscope? Okay, you put optical lens in here. Okay, you maybe put also electrical fields there to replace the optical lens, and you manipulate the beams of lights or the beams of particles and so forth. And this is actually, like what transformer is doing you, or what the attention and the tensor operator doing you, to kind of linearly twist, fold, and merge and multiply, the information. So it's a way of processing information. Microscope is actually maybe the earliest kind of fancy way of processing information, visual information, through many steps of operations, right? And what the biologists saw, in the final step, it's not verified, by the way. We didn't see the real thing, okay? It's based on some kind of, experimentation, on some kind of theory. The theory is not about the physics of the biology. It's a theory about how these informations are processed. And then you build a confidence that, hey, I should trust that. Well, in a sense, what transformer does is exactly that. They are basically doing some manipulation of your textual kind of tokens, or maybe imagery tokens, and then you put there something. If you believe that makes sense, and if you believe that it's repeatable, and if you believe that it is satisfying certain statistical behavior like stabilities and robustness and so forth, you choose to believe it. And then this becomes your microscope. That's the way I see it, right? And therefore, when I put the biological data in there, I do hope that over there, when it is being synthesized and generated in the decoder, maybe we could see biology itself, just like what we see visually from a microscope. Right? So that's the kind of the rationale behind this whole idea about redoing biology using the large pre-trained model. Again, there's a whole zoo of pre-trained models in the space already in text, image, audio, video, and so on. Not yet too many in biology, but they start to appear. For example, AlphaFold is actually a very, very matriculate engineering feast, right? So it basically has some ideas that is unique to biology. For example, in here, there is an input representing the structure. How you use the structure? You use the structure to create now the pairwise distances between all the atoms in your sequence. That gives you a two-dimensional display of the information. It's kind of a way of imaging the protein as a training information. And then the sequence, if you just put it by itself, maybe already informative, but not quite. They put together multiple sequence alignments so that you see, across a species, which part of the sequence is more conserved than others, and that gives you the way to pivot into this matrix to actually weight them differently, right? And then through some kind of, multiple folds of attention integration and layer transformation, you basically are doing the same thing as you do with language using the large language model. And then you can actually make some more empirical steps to recycle the output in here and refine the weights. I think this is just when you train the RBMs or something, you actually have a way to unfold a two-layer RBM into multiple layers of fixed weight kind of propagational procedure. I think this is something similar to that in terms of the high spirit, right? But at the end of the day, you were able to, predict, based only on the sequence about the structure, which is, in a sense, to me, a digital microscope should do, right? And in fact, this is a model built by my colleague Le Song, which is fancier. You replace multiple sequence alignment now with a language model, with a protein language model. Basically, you pre-train that multiple sequences only based on certain ways of tokenization, certain ways of attention mechanism. And then that actually becomes more efficient because multiple sequence alignment is very, very expensive to form during inference. And also, you can only build on a few sequences in alignment, maybe tens or hundreds. But in here, you can train that protein language model with all the sequences you ever have in your database, which is very rich. And they actually gives you functions, like predicting fine structures, predicting interacting structures, and so forth. Right. So that's kind of a roadmap that my team is trying to now push forward. That you can start using this type of technique, first of all, to process a large amount of data, because by definition, these are heavily parameterized models, which needs a large amount of data to be trained. And then also, they are intrinsically able to combine multimodal information. We already see AlphaFold as a combination of a sequence and structures, right? And nowadays, people are working on single-cell models as well, where the gene expression counts over all single cells, but also the gene ontologies, the networks of dependencies of genes or proteins can also be encoded to. It's almost like the equivalent of positional encoding. In other words, here the genes are not positionally encoded per se because they don't have a position. But they have their functional kind of a position inside a network or inside an ontology. You can kind of bring that in. So these models are allowing large biological data and also cross-modal biological data to be naturally integrated. And we ask, what if we just build a bigger enough model that integrates all of that? Wouldn't that be equivalent of modern-day large language model of biology? We don't know, okay? But it's actually at least something intriguing to try if you have the resource. Right? So that's kind of the rationale behind our project. It's not totally crazy, but it's a little bit crazy because we don't actually know what to come out of it. Okay. And well, just to give some confidence of ourselves and maybe to you guys, when we do less ambitious, only single model, foundation model training, we found that whenever we were able to successfully produce it, here are examples of the model we already produced. Some are very big already, a 100 billion protein language model. Some are small, like 100 million single-cell model and so forth. For each of these models, we were able to establish the SOTA results of all the downstream tasks people originally collected sample for. Okay, for example, in biology or in protein, you want to do more than structure prediction, right? You want to probably do the docking prediction, whether two proteins stick together, maybe the affinity prediction, how strong they stick together, right? And also mutation fitness. If you mutate something, do you still see the right structure or not? That's a typical downstream task of protein, and there could be a dozen of that. And in single cell, of course, the trivial thing will be predicting cell types and morphology, but you can actually also try drug response. You perturb the cell by applying a chemical compound or drug. And in the drug industry, this is a big deal. You have to do a lot of wet lab experiment or controlled experiment. But in here, you can also use a model to predict. And the CRISPR perturbation is another thing. You just edit the gene a little bit and see what will come out of it. So what I meant to say that across the 7 models that we trained and across all the tasks that we gathered from the literature to make the performance, we were able to substantially overperform the state of the art, or at least tie with that. And the other performers who are good are also foundation model trained elsewhere. So that kind of suggests that this particular approach at least is showing a good sign of dominating over the traditional approaches, which are based on very task-specific. For example, you train a model to predict the response of gene perturbation. You train another model to do other things. In here, the approach is by definition, multitask and transferable. And also, of course, once the model is given and pre-trained, hopefully, just like what we do with large language model, your customization, your specific alignments of human taste and needs and the safety and so forth becomes a fine-tuning and the prompting. Again, the math is not clear, but it is being practiced with good empirical outcome. In biology, imagine that you could translate that language of tuning and prompting into our personalization of health conditions and drugs and so forth. So in a sense, there is a way to close the loop using, say, a reinforcement learning to do it iteratively and to address many interesting biological problems. So here are just some example of using the model we trained to do structured design. You want a particular antibody to demonstrate a specific structure that can grab the antigen more efficiently. You should probably be able to do that with a totally in silico experiment based on the model. You can also try to model two proteins together to see their interaction and so forth. Right. So just to back to the closing finishing line. So what I just talked about is something different from what many of us, including myself, practiced in the past in machine learning. Here, it is really, from the sequence structure is not using this approach where we have a good equation that allows us to compute everything predictively and confidently, or we have a periodic table that when we mix two compounds, we know actually exactly the chemical behavior of something next downstream. We don't have the luxury. And we can say that, okay, let's focus on a mass and wait for another years before that happen. Well, I don't believe in 100 years we can use mass to compute a structure. Honestly, it's just too complex. But in here, the approach is a completely paradigm shift. You train those slightly more mysterious large pre-trained models, and then you fine-tune and utilize it. And along the way, you perfect it. This is actually how, at least in my opinion, microscope, again, back to that story, was actually practiced. Originally, they were just two pieces of glasses stuck together and see something fancy, and then gets fine-tuned, refined by Carl Zeiss and some other physicists, in fact, who studied optics. And then by the atomic scientists who start change the beam from optical lights to electrons and to other kind of atomic beams, and you actually get more and more magnification power with a lot of good science in that. But the science is not about the subjects. The science is about the vehicle that you get there, right? And those inventions actually have very little to do with biology itself, but biology become the best benefiter of those inventions. And here, we want to see whether with the digital age, with the new foundation models and the deep learning and all that, can biology once again have a paradigm shift away from those old school. Okay, these are some graphs we all know, scaling laws. You can actually even either empirically or maybe theoretically study the complexity of a sample, or maybe providing guideline about how much data and how big a model you need before you kind of reach to the limit. And this emergent property, some of you have heard about, is a kind of still mysterious. We don't actually know what's going on, but we saw those curves already up to a certain size. And given certain data, you see a sudden jump, of some, interesting behaviors and capabilities, maybe due to the inner connection inside the data that we don't know, but picked up by the machines, right? In our recent production of the JACE model, for example, you probably heard it's Arabic, large language model we trained here. It was funny that when the Malaysian president or prime minister came here, and when our team was trying to make a little prep, they found that the language model actually could understand a fair amount of Malay language, even though it's never trained in our model, right? So that's the kind of emergent property, at least I would be curious to see, when we have the biological models, being, trained in that fashion. Okay, just to wrap up, I think we have the opportunity to welcome a new platform or paradigm for the holistic life science in a way different from others, from before, where all this data gets gathered and integrated, and then hopefully solve not just one task, but a whole spectrum of tasks against a single same object, but at different level of granularity, okay, using a holistic model. But again, of course, I don't know how long it will take to get there, but it might be an interesting kind of North Star, to look at it. So to sum up, there's a contrast between LM and what I call the LBM. They have some great deal of a similarity, at least spiritually, and in principle, of course, the actual nuance will be very different. In here, I think the effort that we face is the tedious ones. How to design, for example, the right tokenization of the data, right? How to design the right internal representations, of those latent features, and how to come up with the right architecture to allow information to flow the way we want it. So these are, in some sense, very engineering, exactly like how people design the good old day microscope. But I think this is still, a useful direction, at least for computer scientists. That's basically what computer scientists were trained for. They need to basically make tools, right? So that's where I want to end the talk. But ideally, going back to where I personally came from and the like, I do not see, the rationalism and reductionism, that we practice versus the empiricism and the connectionism that are currently becoming hot. I don't think they are really two parallel paths never meet, right? I think they can cross-inspire each other and provide problems. For example, the model itself could be explained, at least in parts, if we have the right kind of way to look into it. And I don't think also, where the models we see now are really calling for bypass first principles, right? You actually could, maybe design the right experiment to recover the natural laws, based on new findings. So the hope is that these two trends or two schools, can evolve, hands in hand to really meet in midway. That way, in the future, enjoy a new technical paradigm, for biological science that is both, rigorous and, technically, mathematically explainable and provable, but on the other hand, really, hopefully not requiring us to take heavy assumptions and walk away from real life dirty and big data. So with that, I'm done.
Questions for Eric? Yes.
So I'm curious, why did you not keep going in terms of why did you only take biological data, not language data as well, and form one gigantic model that has everything? And where do we draw the line? Because your theory is we need to have these foundation models that understand across all these different domains and holistically. So why not keep going?
Very good question. And I have a very kind of, how should I say, unfriendly, hostile answer, to people on either side. We are talking about, when I talk about biology, or when biologists are talking biology, it's the language of the God. It is basically how DNA, protein, and other things function inside a living organism. The natural language is for human being to consume, to understand, to entertain, to understand that part. Right? If your goal is to understand that and also articulate and communicate, you may need human language. But if your goal is to really uncover the truth, just like many of you like to write equations without bothering giving any explanations, right? So that to itself is enough. In a sense, the language, the human language, the NLP, to me is a contamination to that information because there are a lot of inaccuracy there as well as information. For the information that is already there, they should already also in the biological data. You can rediscover it. So there is no need to get the human part of that. But for the information that is incorrect, they become noise. So if you truly trust there is a need for paradigm shift, say using this foundation model and so forth, why not begin with the clean intrinsic data and leave this extrinsic one out, outside. And then what you can reconcile later if you want to tell a story out of it. You want to create an interface for an individual to manipulate, to produce outcomes by typing human language. Well, that's a utility goal, not the original goal of the model. Right.
I mean, so if I were to push back, I would say that for LLMs, they train them on coding, math, and sciences, and maybe there's a lot of interesting things that could help us
there's
resolve
a
some of the questions, right?
Yeah.
There's logic, and there's structure, and there's mathematics and whatnot. So I don't know. I mean, I agree with what you're saying.
Yeah, this is a philosophical thing. Yeah, you are human-centric. I'm rather nature-centric in this particular case.
Maybe I have a question.
Yeah.
So for these, there's a reason it's easier to believe that having a language model is possible, like understanding language is possible because we as humans do. So there's already like a proof of concept of like a system which can process, create, and understand language. Whereas for biology, no one understands it. Like, so it's not even clear if this goal is even possible. Right?
Mm-hmm. Yeah, that's why I kind of coined the word, the actionable empirical understanding. In a sense, we should humble a little bit, not to say that, hey, only if I understand it, it becomes a truth, right? If it produces something that is repeatable and actionable and creates the results you want, maybe that's okay to begin with. So that's kind of how I take on this approach. Say this AlphaFold thing. I think it's an overwhelming success by all measure, even though people argue against it for certain little tiny accuracy. But it does produce you the structure you want. What else, right? So I would say, yeah, in biology, many of the goals and also the laws, like for example, this law that I don't like is the so-called gene regulatory network. It is a pure artificial thing invented for our little brain, for our very, very poor brain to understand a very complex phenomenon. Then we invent the word network. It is not producing any output because you cannot simulate this network to generate results. So it's for a conceptual understanding of the relationship. If you use that as a bottleneck, you will be stuck in the bottleneck. Right? I've been working on gene network for a decade, and I don't really get out of it, honestly, with anything useful. But if you say, my goal is to predict a cell morphology based on some perturbation, what if I just bypass that network thing, and here I have a black box model, which actually gives you that prediction that you want, repetitively. So in a sense, you are on a different paradigm.
So another question not related to the others. Do you see privacy as being a blocker here for achieving this vision that you're pitching? Because for language models, there's a lot of text on the web, right? Mm-hmm. And we can just scrape it off and train these very large models with trillions of tokens. Mm-hmm. But for this to be able to achieve it, we need a ton of data. And I suppose that data is either not accessible, we don't have a massive data set that has patient records. There's a lot of sensitivity and privacy concerns. Should we let go and just go build that vision?
That's another question I often encounter. People already mix up healthcare versus biology. Okay, biology as the basis. You are talking about a novel, or something fancy out of language. I'm talking about the basis of language. On that biological part, there is no such a thing called patient privacy and everything, because it's pure intrinsic information without a human name attached to it. The human name attached to it is meaningless. All you need is number one, number two, telling them they are different. That's it. Right? So for that data, actually there is, thanks to the very good tradition in biology, every time you publish a Nature and a Science paper, you are obliged to surrender your data to the public. It's been a tradition for 20 years. So many people thought biological data are scarce and not available. That's wrong. Okay, if you look at the PDB database, you look at the GenBank, and there are tons of data out there. It's literally that no one can use it. Okay, the bottleneck is not the data. The bottleneck is that how people can use it right now. There is very few people can use it. Now, clinical data, that's actually exactly the topic in here. When you get a big model, maybe we need to federate it, we need to talk about privacy, that's the next step. You have a pre-trained thing that is based on public data, and you can fine-tune it. You can specialize it through federation or through privacy-sensitive approach. So these are two different problems. They are not together, right? So we don't need actually patient data, his body temperature and so forth, or his address and the wealth and income to train this model. It has nothing to do with that. Biology doesn't need that. Right? So this is actually very important because somehow this whole space has been so contaminated with human perception that we thought we need this and that. The truth is that you don't need it. Okay, I hope I prepare you well for the appetite because it's kind of a crazy discussion. So thank you very much. Enjoy your lunch.